Good evening, viewer. Welcome to our Glory Globe TV. Here's the most amazing news for our viewers. Yes, more than 20 years after the first draft genome from the landmark Human Genome Project was released, researchers have published a draft human pangenome, a snapshot what is poised to become a new reference for genetic research that captures more of human diversity than has been previously available. Geneticists have welcomed the milestone while also highlighting key ethical considerations surrounding the effort to make genome research more inclusive. This is like going from black and white television to 1080 pixels, says Kelly Fox, a genome scientist at the University of California, San Diego. It is something that we have all been waiting for, say Amy Lumka, a geneticist who holds a joint position at the University of Liège in Belgium and the University of Kinshasa in the Democratic Republic of Congo. The current reference genome is missing not only part of the genomic information but most importantly it's missing diversity, he says. The draft genome published in Nature on May 10 was produced by the Human Pangenome Research Consortium, launched in 2019. The international project aims to map the entirety of human genetic variation to create a comprehensive reference against which geneticists will be able to compute the sequences. Such a reference would aid studies investigating potential links between genes and disease. The draft pangenome follows the 2022 publication of the first complete sequence of the human genome, which filled gaps that had been left by the original Human Genome Project. But unlike the original draft human genome and its successor, both of which were derived mostly from DNA of just one person, the draft pangenome represents a collection of sequences from a diverse selection of 47 people from around the globe, including individuals from Africa, the America, Asia, and Europe. Emer Kenny, a geneticist at the Icahn School of Medicine at Mount Sinai in New York City, and her colleagues aligned all these sequences computationally to form a pangenome graph conceptually similar to a London Underground map in which branching paths indicate genetic variation. The researchers found that the pangenome enabled them to identify twice as many structural variants, large genomic alterations such as gene duplications or deletions per person than is possible using the original linear reference genome. The team aims to analyze sequences from 350 people by the mid-2024. Many of the samples being analyzed are from people who took part in the 1000 Genomes Project, a sequencing effort initiated in 2008 to map genetic variation across 26 diverse populations. The participants' frozen DNA samples are being defrosted and reanalyzed by the Pangenome Consortium using a more detailed techniques called long-read sequencing. Yes, this analysis longer sections of DNA at a time compared with older sequencing methods and can distinguish between chromosome pairs from the same person. It is much higher resolution approach, says Fox. Consent forms signed by the participants at the time of 1000 Genomes Project over the analysis of their samples, and during a press conference, Kenny and other researchers from the Pangenome Consortium said that the project was taking further measures to ensure ethical collection and use of the genetic data. For instance, the consortium has committed not to include people who are members of indigenous tribes or other groups that have formal policies preventing contribution of samples, said Kenny. However, some researchers, including Fox, are concerned that the project risks repeating ethically questionable practices from other large-scale genetic diversity projects. For instance, the Human Genome Diversity Project in the 1990s and the ongoing all-of US research program received criticism, including from US-based tribes, for failing to engage sufficiently with members of the communities whose DNA they were sampling. This included people belonging to marginalized groups usually underrepresented in human genetic research. We will, of course, advance knowledge of human structural variation with new data sets and new tools. The progress we should be striving for, however, is the equitable, engagement of underrepresented communities in this work from the ground up, says Crystal Tisoi, a genetic epidemiologist and bioethicist at Arizona State University in Tempe. Tisoi is also co-founder of the Native Biodata Consortium, a non-profit research institute in Eagle Butte, South Dakota, led by indigenous scientists and tribal members. If the research is not benefiting the diverse communities first and foremost, then we are doing something fundamentally wrong here, she says. Beyond the report, Glory Globe TV, Delhi, India.